Human DNA, or deoxyribonucleic acid, is a complex molecule that carries the genetic instructions used in the development, functioning, and reproduction of all known living organisms, including humans. Here's an in-depth look at various aspects of human structure of DNA. Double helix. DNA has a double helix structure, which consists of two long strands that coil around each other. This structure was first described by James Watson and Francis Crick in 1953. Nucleotides. The basic building blocks of DNA are nucleotides, each composed of a sugar deoxyribose, a phosphate group, and one of four nitrogenous bases adenine, thymine T, cytosine C, and guanine G. Base pairing always pairs with T, and C always pairs with G, forming the rungs of the DNA ladder. Genome. Human genome. The human genome is the complete set of DNA in the human body. It contains about 3 billion base pairs. Chromosomes. The DNA is organized into structures called chromosomes. Humans have 23 pairs of chromosomes, 46 in total, with one set inherited from each parent. Genes and non-coding regions. Genes. DNA segments that code for specific proteins or functional RNA molecules. Genes determine traits and play a crucial role in the functioning of cells. Non-coding DNA. Not all DNA codes for genes. Some regions are non-coding but still have regulatory functions, controlling when and how genes are expressed. DNA replication. Cell division. DNA replication is essential for cell division. Cells need to duplicate their DNA before dividing to ensure each daughter cell receives a complete set of genetic information. Enzymes. Replication involves enzymes like DNA polymerase and helicase, ensuring accurate copying of genetic information. Protein synthesis. Transcription. The process where information from a DNA sequence is transcribed into RNA. Translation. The RNA messenger RNA is used as a template to build a corresponding protein. Genetic variation. Mutations. Changes in the DNA sequence can occur through mutations, which can be caused by various factors such as radiation, chemicals, or errors during replication. Recombination. Genetic recombination during sexual reproduction contributes to genetic diversity. Epigenetics. Gene regulation. Epigenetic modifications can alter gene expression without changing the underlying DNA sequence. Environmental influence. External factors like diet, stress, and exposure to toxins can influence epigenetic marks. DNA sequencing. Technological advances. The development of DNA sequencing technologies has allowed researchers to read the entire DNA sequence of an organism rapidly. Personalized medicine. DNA sequencing is used in personalized medicine to tailor treatments based on an individual's genetic makeup. Genetic disorders. Inherited disorders. Genetic mutations can lead to inherited disorders such as cystic fibrosis, sickle cell anemia, or Huntington's disease. Genetic counseling. Understanding DNA helps in genetic counseling to assess the risk of genetic disorders. Ethical considerations. Genetic engineering. Advances in DNA technology raise ethical questions, especially concerning genetic engineering, gene editing e.g. CRISPR-Cas9, and issues related to privacy and discrimination. Understanding human DNA is a dynamic and evolving field, with ongoing research contributing to our knowledge of genetics, evolution, and medical applications. As technology continues to advance, our understanding of human DNA and its implications for health and disease will likely deepen.